This video is about chromosome and the association of chromosome, details about chromosome. First, we will be learning about the nucleosome model, primary level of chromatin organization. The molecular organization of chromatin in higher organisms is best explained by the nucleosome model or solenoid model. This nucleosome model was proposed by Thomas in 1975 along with Comberg and then by Finch. Nucleosome model of chromosome. Nucleosome represents the beads as proposed in the beads on string organization of chromatin. Nucleosomes are the fundamental repeating units of chromatin. The nucleosome consists of a core particle of 8 histone proteins and DNA that wraps around the core. Chromosomes, which are nucleosomes bound to a H1 histone, are separated by linker DNA. Each histone core is encircled by 1.8 turns of DNA. The 1.8 turn of DNA represents about 146 base pairs. The nucleosomes is about 10 nm in diameter. The H1 histone stays outside the histonotamer. Adjacent nucleosomes are connected by a short stretch of DNA called linker DNA. Linker DNA is about 10 to 80 base pairs in length. The H1 histone binds near the site where DNA enters and exits the nucleosome. The interaction of histones and DNA in nucleosome is stabilized by several types of non-covalent bonds. Among these bonds, ionic bonds formed between the negatively charged phosphate groups in the DNA with the positively charged amino groups of histones were very important. Nucleosomes fold to form a 30 mm chromatin fiber, which appears as a series of loops that pack to create a 250 nm wide fiber. Helical coiling of the 250 nm fiber produces a chromatid. Now we can see the structure of a nucleosome. DNA is there wrapped around the histone core. H1 histone is also there. So H2A, H2B, H2A, H2B structure and linker DNA is there. Then nucleosome bead, 8 histone molecules plus 146 nucleotide pairs of DNA. Next is the chromatin histone structure. The chromatin is there, chromosome is there, and linker DNA is there, histone proteins are there, and nucleosome can be seen in the picture. So SMC proteins. The structural maintenance of chromosomes. Proteins are essential for successful chromosome transmission during replication and segregation of the genome in all organisms. SMCs are generally present as single proteins in bacteria and as at least 6 distinct proteins in eukaryotes. These proteins range in size from approximately 110 to 170 kilodalton. And as each has 5 distinct domains, amino and carboxy terminal globular domains, which contain sequence characteristics of ATPase, 2 coiled coil regions separating the terminal domain and a central flexible hinge will be there. So SMC proteins function together with other proteins in a range of chromosomal transactions, including chromosome condensation, sister chromatid cohesion, recombination, DNA repair, and epigenetic slicing of gene expression. So this is the structure of SMC proteins. Head will be there, helical connector, hinge joint, alpha helical connector, and head region. Chromatin is of 2 types, namely euchromatin and heterochromatin. So euchromatin is a less condensed region of chromatin that's transcriptionally active, and heterochromatin is tightly condensed region in the chromosome and it's transcriptionally inactive in general. Then this heterochromatin is again of 2 types, like constitutive heterochromatin and facultative heterochromatin. Constitutive heterochromatin regions are always heterochromatic. Regions that are interconvert between euchromatin and heterochromatin. That is happening in case of facultative heterochromatin. Permanently inactive in regard to transcription in case of constitutive heterochromatin. Next, heterochromatin. When we just try to learn about heterochromatin in detail, the regions of the chromatin that appear relatively condensed and stained deeply with DNA specific stains. It is tightly packed form of DNA. There are two types of heterochromatin, constitutive heterochromatin and facultative heterochromatin. Both of this constitutive heterochromatin and facultative heterochromatin play a role of the expression of genes. Then it's transcriptionally inactive. Even though the facultative constitutive heterochromatin is not transcribing, but it forms a major role in the expression of genes. Then facultative heterochromatin is a result of genes that are silenced through a mechanism such as histone methylation or siRNA through RNAi. Facultative heterochromatin is the result of genes that are silenced through a mechanism such as histone methylation or siRNA through RNAi. Constitutive heterochromatin is mainly repetitive and forms structural functions such as centromere or telomere. Euchromatin. Euchromatin is the lightly packed form of chromatin that is rich in gene concentration. It is often under active transcription. Euchromatin comprises the most active portion of the genome within the nucleus. 92% of human genome is euchromatin. The structure of euchromatin is reminiscent on an unfolded set of beads present in nucleosomes. Nucleosomes consist of 8 proteins known as histones with approximately 147 base pairs of DNA bound around them. The euchromatin, that wrapping is loose so that the raw DNA may be accessed. The first basic structure of euchromatin is an elongated open 10 nm microfibril which is noted by electron microscopy. Then euchromatin participate in the active transcription of DNA to mRNA products. When we just try to differentiate between euchromatin and heterochromatin, first we will be dealing with the euchromatin portion alone. Euchromatin is transcriptionally active. DNA is loosely packed here, actively present in prokaryotic and eukaryotic genome, genetically active, present in inner side of the nucleus, stains lighter, early replicative, are in sticky, allow genes to form a protein, low genetic density, consist about 2 to 3% part of the genome. But now we are going to learn about heterochromatin. When we just look at that, transcriptionally inactive, DNA is highly packed, only present in eukaryotic genome, genetically inactive, present at nuclear periphery, stains dark, late replicative, are usually sticky, regulates genetic integrity and controls gene expression, high genetic density, consists 97 to 98 part of the genome. That's something interesting, like about 97 to 98% of the genome is heterochromatin. Only 2 to 3% of genome is euchromatin. When we just look at the structure of chromosome, we can see that sister chromatids are there, telomeric region, heterochromatin, dark region, centromere, euchromatin, light region, and telomere will be there. So, thank you for listening.